Hi everybody, this is just a short tutorial on the SOMSO brain. So basically what you're looking at is just the base of the brain right here. This is going to be the most anterior aspect of the brain. This is going to be the most posterior aspect of the brain. Right here we have the most inferior aspect of the brain, and then right here we have the most superior aspect of the brain. Now I'm just going to point out some major parts of the brain right here that you can see from this aspect of the model. So this is the brain stem, and then lateral and posterior to the brain stem is your cerebellum. And then this large wrinkled mass right here is your cerebrum. Right here in the middle, this bean-shaped structure, labeled number 46, is your pituitary gland. Now, your pituitary gland is not actually part of the brain. It's actually the master gland of your endocrine system. But it is a handy reference point, and that's why I wanted to point it out to you. So I also wanted to point out two cranial nerve pairs that we want you to know in relationship to this brain. Right here, we have cranial nerve pairs number 1, which are your olfactory nerves. Now, the olfactory nerves are split into two different components. So this long component right here that I'm pointing to with the blue pointer are known as the olfactory tracts, but then the ends of the olfactory tracts that are bowed out a little bit are known as the olfactory bulbs. And basically what happens, and you don't need to know this, but I just wanted to give you some illustration as to why we have these bulbs, what their purpose is, and why we differentiate them from the tracts, is basically what happens is we have axons from sensory neurons of your nasal epithelium, that is the epithelium of the roof of your mouth, and those axons travel through the olfactory foramina of your ethmoid bone. You guys remember that from the last unit, right? And they attach right here at the olfactory bulb. So that's why they're expanded out, is because they're receiving all of those axons from those sensory neurons, which are responsible for your sense of smell. And then that information is relayed to deeper parts of the brain for interpretation through these olfactory tracts. Okay? So we also wanted to point out to you your cranial nerve pairs number 2, which are right here. These are your optic nerves. And your optic nerves actually cross together, I should say more specifically, the axons of your optic nerves cross together just posterior to this point. And I'm going to push down the pituitary gland so you can see that this crisscross point of the axons of the olfactory nerves is known as the optic chiasma, and it's just superior to your pituitary gland, right? Okay, so I think that's all I wanted to point out from this view. So now what I'm going to do is I'm going to point out the structures that you can appreciate with the mid-sagittal view of this structure. And I'm using this half of the brain because this corresponds with the diagram that you have on one of the last pages of your nervous system handout. So again, this region right here is your brainstem. Posterior to the brainstem, you're seeing a mid-sagittal section of the cerebellum. This entire wrinkled mass right here that curves around like an arch is your cerebrum. And then right here in the middle of the brain is a structure called the Diencephalon. Now, I didn't point out the Diencephalon to you when we looked at the base of the brain because it's not as clearly evident from the base of the brain. You can see it, but on the video, I'm not so sure if it would be clearly appreciated. But we would certainly want you to identify the Diencephalon from the mid-sagittal section of the brain. Now what I'm going to do is I'm going to point out some more specific structures within these 4 regions of the brain that I just laid out for you. So now let's go back to the brainstem. So the inferiormost part of the brainstem is known as the medulla oblongata, or just the medulla is fine. This central portion, which bows out anteriorly, remember this is the anterior aspect of the brainstem right here that we just looked at initially, this bowed out region is called the pons. And then the superiormost region, which I'm outlining right here with the blue pointer stick, is the midbrain. Now, the midbrain can be broken up into 2 components, and those 2 components are based on this cerebral aqueduct which travels through it. And I'm going to point out the relevance of the cerebral aqueduct in just a few moments when we talk about the ventricles. All right? So this anterior component of the midbrain is known as the cerebral peduncle. And actually, you have two cerebral peduncles on either side. Basically, cerebral peduncle just means the little foot of the cerebrum. And so the anatomists, way back before they had any idea about what it was actually for, they said, oh, this structure looks like the cerebrum has little feet. And so they named it the little feet or the little foot of the cerebrum. This posterior part of the midbrain is the corpora quadrigemina. Now, corpora quadrigemina, if you break it down, corpora means body, and quadrigemina refers to the fact that there's four of them. So corpora quadrigemina means four bodies. So basically, this structure has four bumps that can be appreciated from the posterior aspect of the midbrain. Now, you probably can only see two bumps here on the model. That's because we cut down these four bumps through the center, so on either side of this brain model, we can only appreciate two bumps. So there's two bumps here, but then over here, this is where the other two bumps are. All right? And again, the corpora quadrigemina is going to be evident for you when you do the, or rather when you watch the videos on the sheep brain dissection as well. All right. So now I wanted to point out the cerebellum. So the cerebellum in its mid-sagittal section is clearly appreciated by these branching tracts of white matter known as the arbor vitae. Arbor vitae literally means the tree of life. And that's the most distinctive feature that we want you to recognize for the cerebellum. All right, so now let's talk about the diencephalon. The diencephalon is probably the most complex anatomical area of the brain. What I'm going to do first is describe the thalamus to you. So the thalamus, which is right here in the center of the diencephalon, is actually two egg-shaped lobes on either side of the diencephalon. You're looking at one of those egg-shaped lobes right here. You see how it's kind of like the shape of an egg? Then what happens is we have this bridge which connects to the other egg, which would be on this model here. So basically what you have is kind of a dumbbell-like structure where the weights are these egg-like lobes, and then here's the connecting point between the two weights. All right? The space here between the two lobes is your third ventricle. And you guys probably know already from lecture that the ventricles are these cavities, if you will, within the brain in which cerebral spinal fluid collects and flows. So this is the third ventricle, a very thin slit-like ventricle, which travels right through your diencephalon and then separates the thalamus into these two lobes. And then this structure right here, your interthalamic adhesion, by the way, I think I pointed that out to you on the nervous system handout tutorial, is that bridge between these two lobes. All right, and by the way, this little pink structure right here represents the epithalamus. That's not a term that we want you to know from the mid-sagittal section on the nervous system handout, but I just wanted to point it out to you nonetheless. You'll probably go over that in lecture. Then, inferior and a little bit anterior to the thalamus, we have the hypothalamus. And then extending from the hypothalamus, and you can see it better over here, here is this stalk-like structure called the infundibulum. The infundibulum then connects to the structure right here, which we pointed out to you before. This is the pituitary gland, the master gland of our body. All right. Okay, so ultimately the cerebral spinal fluid starts being generated in your two lateral ventricles, which are at the deepest points of your cerebrum, and you can't see them on these models. But the two lateral ventricles come together at this very, very thin wall known as the septum pellucidum. Remember I told you before, the septum pellucidum literally translates to the clear wall. Septum is wall, pellucidum means clear. Now there's some little holes, or foramina, if you will. You can actually see it on this model. See right there? Can you see that? See that little hole? You can't really see it on the other side of the model, but there's two of them. They then drain the cerebral spinal fluid which was generated in the lateral ventricles, and take it to the third ventricle. And in fact, still you have cerebral spinal fluid being generated in the third ventricle as well by these little vascular structures known as the choroid plexus, the choroid plexus. And you're going to learn more about that in lecture. That's what this structure represents, is one particular choroid plexus. Plural would be choroid plexi. All right, so they are actually generating the cerebral spinal fluid. So again, the cerebral spinal fluid from the lateral ventricles pours through these little holes. The term for these holes, by the way, are foramen of Monro. You don't need to know that, but they have a name. And then collects in the third ventricle, where more cerebral spinal fluid continues to be generated. And then the cerebral spinal fluid travels down our cerebral aqueduct, which cuts the midbrain into two parts, right? The cerebral peduncles and the corpora quadrigemina. And then it collects in our last ventricle right here, which is this kind of wedge-shaped cavern between our brainstem and our cerebellum. This is our fourth ventricle. And then inferior to the fourth ventricle, we have these little release valves that allow that cerebral spinal fluid to escape out into your subarachnoid space. Little tongue twister there. Your subarachnoid, I don't know why I have such a hard time with that. It's just too many syllables, I think. Your subarachnoid space, which then surrounds your entire brain and spinal cord. Remember, the subarachnoid space is contained within the meninges. All right. Okay, so that basically completes our diencephalon and our septum pellucidum, which is actually more of a structure associated with the cerebrum. We talked about the cerebellum. We talked about our ventricles. What else do we need to cover here? Oh, the corpus callosum right here is this broad band of white matter that kind of looks like an upside down Nike swoosh. Basically, you have millions and millions of axons connecting your two cerebral hemispheres. Remember from lecture that the cerebrum is split up into two hemispheres, right? And the division line is your longitudinal fissure right here, okay? So ultimately, these two halves of the brain are connected through these white matter tracts that compose the corpus callosum. You also have to know that when looking at the cerebral cortex, the upfolds are known as gyri. The singular would be gyrus, and then the downfolds are known as sulci. The singular is going to be sulcus. And I think that's all. And one other thing I was forgetting. Okay, so I'm sorry about this. And technically, we're dealing with another endocrine gland here, so I think it's appropriate to leave it off to the side a little bit. At the very posterior part of your epithalamus, and therefore also at the very posterior part of your diencephalon, we have your pineal gland. All right, your pineal gland, also known as your third eye, is an endocrine gland that's important in regulating your sleep-wake cycles, your circadian rhythms. All right, so that's the last structure that we need to identify there. All right, so that's everything for the SOMSO brain. Again, I think it would be good if you go back to my tutorial where I went over the mid-sagittal diagram on your nervous system worksheet, and then maybe go back to this, stop the slides every now and then. Try to draw out pictures on your own. Do whatever it takes to internalize this material more. I know it's not ideal that you don't have direct access to these brains. I wish I could mail them out to you, but hopefully this will help out. All right, so good luck, everybody, and I will see you soon, probably in some other tutorials.